Hello everyone, and welcome to CK Med. My name is Clark, and I'll be taking you through HIV and AIDS today. I want to shout out to Nikki Shaw for creating the backbone of this PowerPoint, and I've adapted it and added in information that is helpful for your USMLE Step 1. So to begin things off, when discussing HIV, it's nice to know what HIV is. And so it's one of the viruses found within a Lentivirinae. It's a group 6 virus, which is your single sense RNA positive sense retrovirus. HIV specifically has 2 copies of this single-stranded RNA positive sense in its genome. And the structure, the one that we want to particularly emphasize, is the D type, which is that cylindrical nucleocapsid core. They like emphasizing that for exam purposes. So symptoms are, it can cause neurological disorders in addition to immunosuppression. And this is caused from HIV-1. And most specifically, the most common form of this is actually group M, subtype B. That is the most common. HIV-1, group M, subtype B is the most common found worldwide. And then HIV-2 is another virus found within this group. There's other subtypes besides B, such as C and E. These are transmitted heterosexually, and E is transmitted more between sexual partners than is the most common around the world, which is B. Subtype D is another one that is more associated with vertical transmission than most of the other subtypes. And so that's kind of more specific questions. Just someone comes with HIV, they have subtype D, this is most likely how. And they're probably talking about an infant that has vertical transmission. Or if it's C and E, it's maybe something like heterosexual or sexual partner. So what are retroviruses? So retroviruses are viruses that have RNA genome that once they enter into cells, they take their RNA genome and turn it back into DNA genome, and then that can be incorporated into host cells' genetic material. And so there's 2 types of retroviruses that you should understand. One is a simple retrovirus. So this is something like HTLV is a great example, which is human T-cell lymphotrophic virus. And this is not HIV. HIV is a complex retrovirus, not a simple. So simple retroviruses have a GAG, which is a group-specific antigen, which is like the core and capsid proteins. It also has a Pol gene, which creates your polymerase, such as RT, your protease and integrase. And then it also has an M for envelope or glycoproteins. And that's important for those proteins for attachment and reinfecting different cells once it buds off, And complex retroviruses, such as HIV, have these same genes, the GAG and Pol and ENV, but they also have accessory genes such as your TAT, REV, NEF, VIF, and VPU. And one thing that you should definitely know in complex retroviruses is that they actually have more open reading frames than simple ones. So they actually have 9 open reading frames. And then within those open reading frames, so pretty much you have a gene. Say you have a genome here, and it's a really long thing right here. And we would have a promoter sequence here, and then we'd have our polymerase to come in, and they would continue down here. Either something to make our genome from, or to make complementary strands, and then that could be going to make like protein or something like that, to go find some ribosomes. Anything in this process, we have this promoter and primer kind of promoter region over here that we start. Well, this is known as a reading frame, this opening sequence here. Well, how about we open it in multiple regions, and this this is what we call multiple open reading frames. And so these are different areas that our polymerase can come and read these different genes and just produce those proteins or those kind of subsequent matching RNA pieces here. And so this actually, in complex retroviruses, have 9 open reading frames, and then that leads to a production of 15 proteins. So how do you get from 9 reading frames to 15 proteins? Well, obviously it makes sense that you're making polyproteins that just get cleaved into smaller protein pieces, and then those can function as multiple. So say I have my first open reading frame that makes 3 proteins, which totally makes sense as how you can get to 15. And so this is something, and the difference between complex retroviruses is that you have more open reading frames and you have even more proteins. And you should know this 9 and 15. This is actually pretty high yield for exams to know specific for HIV. It's these small details that they like for this guy. And do you know them, kind of? What happens is once we get our virus inside, we create what is known as a provirus. So we form DNA from our RNA template, and then that integrates into the genome, and this is known as a provirus. It is not a prophage. Prophage is when you have a virally infected bacteria, and that's why we call it a phage. When it's a virally infected human cell, we call it a virus, and so that's known as a provirus. And pretty much this provirus is the template for synthesis of viral mRNA for the genome and for the mRNA to make their proteins associated. So then we can kind of put proteins and we can put genomes in here, and we can have other proteins like our RDDP, which is our reverse transcriptase. And we can package this out, make an envelope over here with our little associated envelope proteins. And then we have, look, an HIV virus over here, and that can be secreted. And then we also have this viral mRNA. So this is a non-structural, and viral structural proteins can come off of this mRNA protein. So this is everything I just pretty much have discussed. So HIV, so where did this come from? So the origin, it was discovered in about 1983, and there were 3 theories as to where this had come from, such as contaminated oral polio virus or vaccine in Africa, even smallpox vaccine contamination, or that it could have been acquired from non-human primates directly. Because there's a simian immunodeficiency virus, or SIV. This is why we were thinking maybe it had originated from that virus and then had progressed into what we know now as HIV. Remember, group M and subtype B is the most common, and that is the most common of global epidemics. So transmission, how do you get HIV? You can get it from blood, semen, vaginal fluid, so unprotected anal, vaginal, or oral sex, you can get this through sharing needles and syringes. Vertical transmission through the placenta, and male circumcision is protective against this, as it is for many carcinomas, such as involving with papillomavirus as well. And I just put a little syringe down here. It's a little Lego syringe. I wish I had one of those when I was a kid. All right, so HIV proteins. So these are actually really important to know, each of them, especially the first 4 that I have emphasized here. And so what happens is, we talked about some of the genes that HIV has, and one of them is your envelope, or ENV protein. So your envelope proteins pretty much is associated with making proteins that are going to be bound and hooked into the envelope, allowing for further infection to take place. And so what are these glycoproteins on the surface? And these are your GP41 and GP120. But first, what we need to do is make a big glycoprotein. Then we need to cut it into 2 small pieces. So that big glycoprotein is known as 160. So that is actually what is straight produced from your envelope protein. So remember how we talked about reading frames? We make a protein from here, and then we chop that protein into multiple pieces. This is how we get those 15 proteins from the 9 open reading frames. This is an example of 1 that makes 2. So we have envelope gene that produces envelope protein, which is your GP160. Cut it into 2, your 41 and 120, because scientists really love math, and they know that 41 plus 120 equals 160. Just so you know that, in case you thought it was actually 161 from your math training in elementary school and high school and college and just general life, you've actually been, you lied to all your life. It actually is 160. 41 plus 120 is 160, just so you know, okay? Don't make that mistake, okay? We're in medical school. We're now smarter now. We don't want to make that simple math mistake, okay? So GP120, this is actually the surface component of the protein. So we have this emphasized up here in the light green here. And this is actually what binds to the CD4 molecules of T cells. And this is actually important as far as its pathogenesis. Also, we have our GP41, which is kind of the thing that hooks GP120 and holds it into the membrane as a transmembrane protein. But it also is important for fusion of that membrane. So what happens is we kind of bind to that CD4, we bind to co-receptors, which we'll be discussing, And then we bring the membranes a little bit closer. And this GP41 allows for the direct fusion of those membranes together, so then this virus can enter. And so what we can do is we can actually target this GP41 and block that membrane fusion altogether with Inflavatide. And so that just makes sense that infusion, or Infuvo, is blocking infusion or fusion. And GP, or I'm sorry, not GP, but P24 is the capsid protein that kind of surrounds here. So this is your purple capsid protein, and this is your nucleoid capsid. This is your conical shaped thing that I was discussing. And this is actually what we search for on PCR. So remember when we talked about hepatitis B virus infection, and so when someone is infected with hepatitis B, we look for antibodies against surface, and we look for antibodies against capsid. And so that anti-C, right? So anti-HBC antigen antibodies is something we look for. And when you see both of those positive, that means this person has been over they're over their infection, but it actually was an infection. It wasn't just our vaccination. Remember, vaccination was just the surface molecule, but if you have an antibody towards the core as well, or this capsid protein, then that's something that to say this person had an infection. And so this P24 is actually equivalent to that C of hepatitis B, that core protein. This is the capsid protein. So this is known as P24, and this is what we look for on PCR. And so we want to see if the cells are actually producing this. We can also find antibodies against this, and this is something that tells us pretty much this person is infected with HIV. This is our confirmation test. So that's why we look for that guy. Also, P7 and P9, these guys make up the nucleocapsid protein, which just surrounds the RNA found within here. Remember, when you have single-stranded like RNA or DNA, it's not very stable. RNA is a little bit more stable than DNA, however, but it's still not that stable. So we want to kind of cover it up and protect it, and that's with our little nucleocapsid proteins, and that is P7 and P9. We also have one last protein I want to kind of quickly mention here, which is your P17. And so P17 is just important for your it's actually a protein found right here, this really small one right here at the tip of here. And it's only pretty much important as far as a matrix protein. That's these blue things right here. See all this blue around here? These are just matrix proteins that kind of holds everything all together and keeps the membrane's shape intact for the virus to infect optimally. And that's what our P17 protein is for. All right, so now on to the life cycle. This is actually really, really important. You need to write this out a couple times, and I have some helpful ways to remember some of the aspects of this. But this is actually super important for you to understand, because there's going to be a lot of questions that come from this. As far as HIV. HIV is one of the most high-yield things of microbiology that you'll have because it's so common, it's so rampant, and we need to know how everything works. Because if you actually learn through this, learn all of these steps and how it works and how we kind of put our drugs and treatments for this, when you get to farm, it's going to be no problem. It's going to be so easy for you. So I want to emphasize good ways of learning this, and so you can kind of cruise through this section in farm. So the HIV life cycle starts with binding and fusion. So I talked about GP120 and how that binds to your CD4 molecules found on your T cells. And once it binds those, pretty much what it needs extra is a co-receptor. And we're going to talk about those co-receptors in just a second. And once it binds those co-receptors, this kind of double binding allows for closer interaction between kind of that capsule or that envelope of the virus and the membrane of the host cell. And this close proximity allows for GP41, which is that kind of transmembrane protein, to do its job. And its job is pretty much to cause that last step in fusion of those membranes together, and that allows that RNA or the RNA pretty much the virus to enter into the cell, and that releases the RNA into the host cell. And so these co-receptors that bind actually kind of depend on the stage of infection you have. And so when you have a naive infection, so someone that just got stabbed with a needle at a hospital, and that needle was recently in someone that had HIV, and then they got pricked with it, and so we need to start them on treatment. But however, this infection in them actually is first going to be used utilizing the co-receptor CCR5. So this is found on T cells. It's also found on macrophages. And macrophages are a huge aspect of the initial infection of HIVs because HIV goes and it lives in macrophages, and macrophages take it all over the entire body and just spits it out all over in our lymph nodes. Remember, macrophages, when they phagocytize stuff, they go, okay, I need to bring this now over to my lymph nodes. So they bring it over the lymph nodes, and they go, hey, look what I found. Well, this is a problem because when they say, look what I found, lymphocytes, like, hey, look what I found, T cells. Our T cells, don't bring that to our, and then the T cells get infected. So as far as how does it get in macrophages, it uses this CCR5. It also infects those naive T cells via this co-receptor. And how we can block this, so we can actually block CCR5 specifically with Maraviroc. This is a co-receptor blocker as far as stopping the initial infection. And then we can also have, once you have had this infection for a while, HIV actually utilizes on T cells a different co-receptor, and this is known as CXCR4. So I remember this is when you've crossed this, or X'd this, before, so CXCR4, when you've crossed this before, meaning that this is a later stage in this infection, then you use that CXCR4 as your co-receptor. And this is so high yield, and I was like, oh, this is a free point with that little mnemonic right there. It just says, oh, someone's had HIV for a while. What co-receptor are they using? Well, they've crossed it before, and so they've had it for a while, and that's CXCR4, which is nice. So the next stage that happens is once this RNA has kind of been spit into the cytoplasm or plasma, there's also a protein that comes along with it, which is your reverse transcriptase. This was found within the nucleocapsid along with the RNAs. And what this does is now is active, and it converts single-stranded RNA into double-stranded DNA. This is known as a reverse transcriptase, or your RNA-dependent DNA polymerase, RDDP, RDDP. Okay, so that is your reverse transcriptase. We can inhibit this with two classes of drugs, your nucleoside reverse transcriptase inhibitors and non-nucleoside reverse transcriptase inhibitors. These guys actually bind the enzyme itself in an allosteric site and stop it from working, and then these guys actually kind of inhibit the chain elongation of these guys, of the RT. And so what happens if you use reverse transcriptase, and actually this is high yield for micro, when you stop reverse transcription, that stops RNA into DNA, so therefore you're going to back up your RNA in the cytoplasm. So be looking out for viral single-stranded RNA in the cytoplasm, and that is when you're stopping reverse transcriptase. Where you also have the next step, once you have that double-stranded DNA, you actually integrate this into the genome. And so this newly formed DNA enters the host cell nucleus and then is inserted into the host DNA. And this is using an enzyme known as integrase. So this integrase comes along and is produced from the virus itself. And so what we can actually do is we can target this enzyme, which is nice. This is known as integrase inhibitors. This is another class of drugs. And if we block this, we have converted it into DNA, but we can't put it into the genome. And so we have a backup of viral DNA, double-stranded DNA in the cytoplasm. And that's what we can find. We can also find it in the nucleus as well. So just a backup overall in the cell because it's not being integrated. And then next we have transcription. So we have a provirus. Once it's been integrated, we use the host RNA polymerase, and we copy the HIV genome and mRNA, and that helps us to kind of make those proteins. Remember that this can also remain dormant in there for many years, and so this is why this infection isn't always very rapid. Next is assembly. So now that we've produced some of the proteins and everything like that, we actually are able to use some of the protease to cut up some of the longer chains of HIV proteins in order to spit them out over onto the cell membrane, and that allows for those glycoproteins such as GP41 and GP120. Those guys are cut up from this protease, and that is found on your envelopes. And then what we do is we kind of put everything in a big sack and we bud it off the membrane of the host cell's outer envelope. And that is where we get our envelope for this virus is the cell membrane. And then lastly, once it buds, then we will mature the full virus. So here we use the protease. We cut some of the long chains of the HIV proteins into smaller individual ones, but it wasn't actually in maturing some of the proteins. So we still have long proteins involved, and this HIV protease cuts it after we bud. This is known as maturation, and this then gathers everything together and forms the capsid and the nuclear capsid around the genome, and then the reverse transcriptase kind of hides out in there, and we can actually block this with protease inhibitors. So the protease is that thing that cuts all these things, and that is everything, all those drugs that end with Navir, and so you never mess with a protease inhibitor. That's how you remember that guy. So pathogenesis, we a little bit talked about this. Remember those macrophages and those T cells, and infects those. It also uses another cell type known as dendritic cells. So macrophages, we replicated within there, and then they brought it to lymph nodes. Dendritic cells bring it to lymph nodes, but they don't actually have them infecting. They actually just accumulate them on their surface. They don't internalize them, and they just carry them to lymph nodes, and then great, we now infect our CD4 cells. Thanks for bringing this to us in the lymph nodes. Wonderful dendritic cells, ding nuts. Sheesh. So in immune evasion as well. So this is actually super high yield. That is super weird because HIV is just a horrible virus. Retroviruses just make tons of mistakes all the time. So there's constantly tons of different changes in the genome, and there's a bunch of just false protein or like virions and stuff that they make. And so some of the things that they can change, because they make so many genetic mistakes, they can make antigenic variation, so our immune system doesn't respond to it properly. It also has carbohydrate masking of like target epitopes. This is actually super high yield, this point right here. It also can have conformational changes in the viral envelope, downregulation of the host HLAs, and then latency and dormancy in T cells and APCs. These guys are all high yield. Make some flashcards. Don't miss that question on your exams. This is something you definitely need to know for HIV. It's like, why is someone continually having this infection? Why can't our immune system respond to it? I know it's infecting our T cells, but still we should be able to get rid of it at some point in time, but we never do, and it's because of this evasion. So cytopathic effects. So HIV is something that can cause syncytia, similar to our herpes virus. And those actually cause fusion of macrophages, which is very weird, and microglia in the brain. So this is seen in the brain. This can cause encephalopathy and dementia as symptoms that come up with this guy. And then we also have this spreading from cell to cells in immune tissues and everything like that. And so our immune antibodies pretty much are ineffective against HIV. And so that's when we get to this guy, we can actually talk a little bit about that. So after the acute infection, we'll be talking about what the acute infection entails, but it's nice to understand how is serology working, how are the virus replicating, and everything like that. And you can get a nice, good picture, solid picture of how HIV kind of works. So in the acute infection, we obviously have cells virus replicating. So we see this massive rise in our P24 antigen. So this is similar to hepatitis B. You know, we have that high rise in that surface antigen, and that's just saying we have replicating virus. So we have a high response to this. Then what happens is, remember that these guys as viruses replicate internally, and so they're no longer floating around the blood or lymph. They eventually get picked up from macrophages and dendritic cells, and they're living inside of T cells, and they're staying in the lymph. So I'm not really, this is a blood test that I'm gathering all this data, so I'm not really going, oh, let me suck out our lymph nodes. It's actually just let me suck out someone's blood and figure out what's floating around in there. Well, all these viruses are found hidden in cells. They're almost like latent. They're starting their latent infection. So that's why our P24 antigen no longer is really kind of floating around. It stays latent, and that's why it's in a low concentration, and that's important. But it's also because not only is this cell these replicating virions latent, or just replicating and staying within the cells and not really popping out, and they're actually going from cell to cell causing syncytia, but there's still P24 floating around. It's still spilling out into the blood, and so we should be able to pick it up. However, remember, after a certain amount of time, we create antibodies. And these are anti-HIV antibodies, and they bind that GP120. And also we have P24 antigen binding specific antibodies. And both of these kind of rise in serum, and they neutralize and bring out. They kind of remove all this P24 and this GP120 antigen that's flying around all over the place. So that's why this also decreases here. It's not just because we no longer have this P24 flying around because our cells our viruses are inside of cells hiding and not spitting it out into serum, but it's also because we're neutralizing it with some of the antibodies. In addition, another thing that's found on here, we also see our CD4 count decreasing. And this is obvious because we're causing an infection in our CD4 cells, and so they're dying off, and that's why their titers are going down throughout the progression of this. But you might ask, why is there this peak and then decrease and then peak and then kind of decrease overall? So pretty much what happens is we have macrophages and dendritic cells saying, hey, look what I found. And so our T cells are going, hey, look what you found. And they're responding. And so they're proliferating and cells are going and responding to an infection, similar to any infection we would find, right? They're getting invaded in the process, obviously, but they are responding and they are proliferating as if it's a normal upper respiratory tract infection or something. We're going to see an increase in our T cell count. However, it goes down because a lot of these cells are dying, right? They're responding, but when they respond to the scene, they get killed. And so that's actually what is happening here is they're proliferating, they're responding, and then they're dying. And that is how and why these peaks are found within this graph. So as far as the symptoms and how this comes up, so acute retrovirus syndrome, this is the first stage that we talked about. This is a flu-like symptoms. It's kind of masked and very similar to a lot of infections. It's severe sore throat and fever and mono-like symptoms. So when someone has a flu-like symptoms, it's nice to get at least a nice brief sexual history to figure out, are they sexually active, homosexually active, or something like that, or do they do drugs? Do you do IV drug use, right? That's why we ask them in history, why do you do any recreational drugs? Do you take heroin or anything like that? Do you do IV cocaine or whatever? So those sorts of things we need to be asking our patients because that gets a nice picture and helps bring in diagnoses such as HIV. In term 5, you're going to get a case of a patient that has HIV, but pretty much they come in and say, I have a sore throat. So you're thinking, oh, it's a head and neck exam and everything like that. And if you're not asking proper questions, you will never get the diagnosis of HIV. This person has acute infection of HIV, be looking out for those severe sore throats. Then what happens is after we have this seroconversion, we start producing antibodies. We're going to have this window period where our HIV test is negative, our antibodies are negative because they're kind of neutralized. And this is similar to like hepatitis B infection. However, we can look for viral load specifically, and that actually will be positive, and it will be very, very high, and that's done via PCR. And this is something that we want to do. This is part of our 4th gen ELISA, is we check for antibodies, and if they're in this window period, it'll be negative for antibodies. However, we can check viral load, and those will be high, and that'll be a confirmed test that this person actually has HIV. So we do those things kind of simultaneously. Also, later stage, we start seeing opportunistic infections, which is found in the later stage when our CD4 count starts dropping, and that's when we're more associated, again, with those less than 200, sometimes less than 500s. You can see things like candida infections and such. And then once we start seeing opportunistic infections, we now can classify that as AIDS if our CD4 count is above 200. If it is 200, even if there's no opportunistic infections, we still call that AIDS. So less than 200 AIDS or greater than 200 and opportunistic infections. So treatment for this, we talked about these a little bit as far as incorporation to the stages of their life cycle. And so these can be your non-nucleoside reverse transcriptase inhibitors, or NNRTIs. These guys are specifically ineffective to HIV-2. This is very high yield for pharm, in addition to it could be asked as a question in micro as well. NRTIs, nucleoside reverse transcriptase inhibitors, are our kind of go-to first line that we use. Protease inhibitors, we do use these as well. Fusion inhibitors, CCR5 antagonists, such as Maraviroc, and our integrase strand inhibitors, which we like using as well, which is your INSTIs. So now let's talk a little bit about AIDS and some of the infections that come from this. So AIDS, like I was saying, is a CD4 count less than 200 or the development of opportunistic infections. This is very important for you to understand. So things that come upon are candida, fungal infections, all those sorts of things, and we'll be discussing each of these in later slides. And then progression, pretty much we have discovered and talked about. So some of the bacterial infections that are associated with AIDS that you should know, pneumonia is very common. So just think of the most common community-acquired pneumonia, and that is Streptococcus pneumoniae. Now, we know this is just a pathogenic organism, and it can cause pneumonia in me. I don't have AIDS, but it can cause pneumonia in general population. However, just think of how worse it is in someone that has AIDS. So obviously we need to bring this up. This is important. This is a common cause of death in patients. It's not the most common, but it is a common cause of death in patients. And so these guys are kind of colonization of the oropharynx. Remember these virulence factors like IgA protease, and it has a pneumolysin. And then it stimulates inflammation with that pneumolysin and kills it. In addition, it has things that would stimulate it, such as teichoic acid and peptidoglycan, or peptidoglycan. And it also evades the phagocytes through that polysaccharide capsule. Remember, our sickle cell patients or patients that don't have spleens are more prone to these types of infections, but also AIDS patients because we don't have an immune system to respond to it properly. It also causes severe pneumonia and dissemination. And remember, when you have strep pneumo dissemination, meningitis is common. So be careful and looking out for those guys, right? And then another infection that's super important is your Mycobacterium avium complex, or MAC complex. And this is about 20 to 30% of AIDS patients actually have this. It's CD4 count less than 50. This is the only thing you should know for CD4 count less than 50. And it can cause a solitary nodule, acid fasting, but however it is TB negative, so it will be a tuberculin negative test. And I actually want to reiterate this CD less than 50. They like asking what is the specific 50 for this guy. But remember, once you're less than 50, all the infections that we're about to talk about can show up, right? So it's literally starting to include more and more as your CD4 count decreases. But this is something that they like to point out in stems, and it's more like a TB-like syndrome, but it's TB negative, and that is going to be your MAC complex. So it can cause disseminated similar tuberculosis and lymphadenitis in children and stuff like that that might have AIDS and those sorts. Tuberculosis, so this is actually the leading cause of death among patients with AIDS. It is not the most common infection. It is the most common death-causing infection in patients with AIDS. Salmonella, it can cause kind of chronic bloody diarrhea, vomiting, fever, recurrent episodes. It also can cause Reiter syndrome, which is conjunctivitis, urethritis, and reactive arthritis, which is kind of rare for GI infections from Salmonella as well. Bartonella henselae, we have talked about this as your cat scratch disease, so it can cause regional lymphadenopathy that is chronic and it stays there. That is chronic cat scratch disease, but it can also cause tumor-like growth, which is known as bacillar angiomatosis. This is a vascular proliferative disease. It's very, very similar to Kaposi sarcoma, but it's caused from Bartonella henselae. And so it looks bright red skin patches, very similar to Kaposi's. However, how you differentiate it is based on H&E staining. So remember, this is a bacteria. So what do you think responds to bacteria normally? Well, neutrophils. So that's how you know that this is bacillar angiomatosis, where Kaposi's is caused from HHV8, which is a virus. So we're going to have lymphocytes responding to that guy. So now on to our viral infections. So viral hepatitis is actually quite common among patients that have HIV/AIDS. Why? Because HIV and hepatitis B and C can be incorporated with IV drug use. So these are patients that are high risk of contracting all these illnesses. And if you have hepatitis B and C and HIV, you're not going to be fighting off those infections. You're going to have chronic liver damage, liver cancer, cirrhosis, and you can have an increased risk of liver toxins such as alcohol in production of acetaldehyde and acetaminophen in production of your NAPK. You can also see decrease in like HBs, HBC antibodies because now our immune system is not responding. You lose your memory T cells. If you lose your memory T cells, you lose that stimulation of your B cells that were producing the antibodies, and so you're no longer producing those HBs and C antibodies. So cytomegalovirus is another guy. Remember, we mentioned that this is when it's less than 100. You can get that cotton wool retinitis, and that can cause blindness. And then GI esophagitis, we've talked about in the GI section, and respiratory tract like pneumonia and such. Remember, this is also associated with kidney transplants, and that is high yield to no. Human herpes virus, HHV1 or 2, also known as herpes simplex virus or HSV1 and 2. This is usually worse as CD4 count drops, which really any infection does, but they emphasize that viral shedding increases when you have CD4 count that decreases. And this causes sores and like cold sores or genital sores that last a lot longer, and they stay there, they're persistent. Also, this can cause meningitis for HHV2 or HSV2, and encephalitis in HHV1 or HSV1, and this is very bad because it causes brain damage and blindness. And this is reactivation encephalitis. You're going to talk about this more in the CNS section, but this is something that causes reactivation of encephalitis. You see something that's describing that in a stem, I want you to be thinking of this actually first before most of anything else. And then dissemination could be very, very life-threatening and very dangerous. This has come up on exam questions, so definitely know that disseminated HSV1 or HSV2 plus HIV infection and AIDS progression is very, very bad with these, or very dangerous. Human papillomavirus, this is more associated with women's cervical cancer, increased risk because we're not fighting off that virus anymore because our T cells are dead. So that makes sense, right? And progressive multifocal leukoencephalopathy. This is caused from a polyomavirus known as JC virus, and this is when you have a CD4 count less than 20. Similar presentation, similar to multiple sclerosis, except it kind of stays for a while. So this is like hemiparesis, unilateral blindness, and sensory loss. It's not like it doesn't come and then go similar like multiple sclerosis. But also can cause speech problems. So someone with weird neurological problems plus HIV, or they were an IV drug user, be looking for this. Now onto some of the fungal infections that are associated with AIDS. So Candida can cause mucosal and dissemination. I think beating a dead horse with this. We brought this up multiple times, but oropharyngeal less than 500 and esophageal less than 100. Vulvovaginal infection can also be associated with AIDS, but it's associated with general yeast infections as well. But also, Candida can cause pneumonia and dissemination. It's more rare, but it is more associated with AIDS than anything else. And that is your Candida albicans. And then Cryptococcus, this guy is actually kind of interesting. So when your CD4 count is less than 100, you can have fungal meningitis caused from Cryptococcus. So remember, this is your only encapsulated or polysaccharide capsule yeast. It's only encapsulated yeast. You get it from soil and bird and bat droppings, and produces this enzyme known as phenol oxidase, and we can actually test for that. In addition, it grows at 37 degrees, and we would do our India ink staining on CSF extraction when we do a lumbar puncture, but we can also do our mucicarmine stain for that capsule to see as well. And you'll be more introduced to this and the details of its badness in the CNS section later on. Pneumocystis jirovecii, or PCP, or pneumococcal or pneumocystic pneumoniae, which is PCP. This is your CD4 count when it's less than 200, so just the general AIDS criteria for pneumocystis. This is actually the most common infection found in AIDS patients, the most common infection. It leads to death in some patients, but not always. We can also just treat it, and it's not super problematic, but it is the most common. TB is the most common death-defining AIDS illness. So that's important to know those guys. So it causes interstitial pneumonitis. And plasma cells kind of respond to this, and we can use antiretrovirals to pretty much prevent this from accumulating in this patient's lungs. And it's usually almost exclusive to HIV and AIDS patients. You don't see it in anybody else. This can be causing a non-productive cough. It's dry, nothing comes up, and this is that candy cotton exudate on histology. And we'd be looking for cysts and trophozoites. Even though this is a fungus, it doesn't respond to antifungals because it does not have ergosterol in its membrane. And usually we target ergosterol synthesis or ergosterol itself specifically as far as fungal infections, but this guy doesn't have it, so we'd be doing nothing with antifungals. And so we'd actually have to treat it with cotrimoxazole, which we actually use for urinary tract infections and stuff like that. This is actually a combination of two drugs, which you'll learn in pharm. So now some of the parasites that are associated with AIDS. So plasmosis, this is Toxoplasma gondii, is the most common CNS infection with AIDS. And it can lead to encephalitis, like headache, confusion, motor weakness. It's spread via cats and their poop. And so just think, crazy cat ladies, or think old cat ladies, what happens is on CT, we can actually see ring-enhancing lesions. So there's actually a few things that we can see ring-enhancing lesions in the brain. You're going to come across, in the CNS section, you're going to talk about ring-enhancing lesions such as an abscess. And that's usually staph or something like that. That can cause that. It's usually one lesion. Now, if you see multiple, toxoplasma should be popping in, especially if they're AIDS patients. AIDS plus multiple ring-enhancing lesions, you're thinking of toxo as the top of your list. Right? Other ring-enhancing you're going to learn about in PATH. Some tumors actually have ring-enhancing lesions, such as your glioblastoma multiforme can have a ring-enhancing lesion, but that's usually very large and crosses from one hemisphere to the next. That's your butterfly lesion, but remember, it is that ring-enhancing lesion as well. So Cryptosporidium parvum. So we talked about this in our GI section. This is a very common cause of kind of persistent diarrhea in patients. This is associated when you have CD4 count less than 100, and it's due to contaminated food and water or swimming pools and stuff like that. It's very common in children. It's the most common cause of chronic diarrhea in children, and it causes dehydration, malnutrition, cholecystitis in HIV patients, and it can be could be self-limiting in HIV patients when they have a CD4 count greater than 200, but when it's less, it's a problem. We can also do acid fast staining of those. Remember what those look like? Go back to those slides. They're little pink little circle guys. Another guy we talked about in our multi-systems and our skin is leishmaniasis, and it's often associated with the visceral type in AIDS patients. And remember, that's with the sandflies. The sandflies out in the desert, and they're on leashes. It's a leash mania. Oh my gosh. Okay, usually find this in Bangladesh and Brazil, Ethiopia, India, Sudan, those places. You'll have pretty much all these symptoms down here, including hepatosplenomegaly and anemia. Now on to the cancers and neurology. This is one of the last things we're going to be discussing in this module. Kaposi sarcoma, which is caused from HHV8 or human herpes virus 8. This is very high yield to know for exam purposes. And this is pretty much a tumor of blood vessels. And we have to differentiate it from bacillary angiomatosis, caused from Bartonella henselae, which is your cat scratch disease. Why? Because they look very similar presentation to one another. And so pretty much what you got to do is look at H&E of one of these guys, and this is what's found in this guy's gums right here. It's kind of gross. And those are lymphocytes that are responding. So this is a virus inside cells, and you think what responds to intracellular organisms, and that's going to be your lymphocytes. The one exception, I actually want to bring you back to that, just a little bit of reminder, kind of tie-in to lymphocytic response. So there's actually one bacteria that you should know that lymphocytes actually respond to that is extracellular. You can think of that. I did emphasize this when we discussed it in respiratory section, and it causes a cough that is severe in children, that it gets to the point where they cough so much that they vomit, and hopefully you're thinking of whooping cough. And that is actually the guy that causes a lymphocytic response to an extracellular bacteria, which is very abnormal. And so we kind of think that, kind of associate that with more of a viral infection, but we would be wrong. So that was just a little bit of tie-in back into respiratory. Well, any reminders we can get is good. These are pink, red, purple lesions, as you can see, and this guy's face, or his gums. So other things are non-Hodgkin's lymphomas, and they originates in lymphocytes within the lymph nodes, but they also can be in lymph tissue found in the brain. So if you have an HIV patient with neurological symptoms, we look at CAT scan and we see a tumor growing. Be thinking, this is most likely a lymphoma, a B-cell lymphoma of the brain, or in the brain. This is very high yield. This showed up on both micro and path questions, so definitely be keeping that in mind. Also, it increases risk of other viral infections and stuff like that, such as HTLV, hepatitis C, and EBV. Guillain-Barré, remember I mentioned that HIV can cause Guillain-Barré, so I just wanted to reiterate that as well. And lastly, our last syndrome that we want to talk about is IRIS, or Immune Reconstruction Inflammatory Syndrome. This was kind of confusing. They weren't really good at explaining what the heck was happening. And so I hopefully can describe this so it makes sense to you. So they described it as a paradoxical worsening of an infectious process followed by HAART and in HIV patients. So that doesn't quite mean as much unless you understand what is happening. So remember, our HIV infects our immune cells drop a number. And so we have no response to infection. So we get opportunistic infections such as TB and MAC and pneumocystis and all those things like we have just been discussing in the last 10 minutes or so. And so these patients have these underlying infections, right? TB, MAC, pneumocystis, cryptococcus, all those things, right? And so our immune system's not responding. So they're not having symptoms really associated with it as much as someone that has an immune system. Why? It's because remember, back in immunology, you learned about immunopathology. So take influenza, for example. Why, when you have influenza, do you have a cough? Well, that is straight up damage to your respiratory epithelium because of the virus replicating in it. That totally makes sense. But why do you have muscle aches? Why do I have leg muscle aches and chest muscle aches and arm muscle aches and neck muscle aches when I have influenza? Well, there's no virus in my blood all over the place. That would be really bad. It's actually your immune response, your cytokines that are secreted in your immune response cause all that myalgia and fever and pain that you're having, and that is why this is an overall syndrome of influenza virus, right? Well, here these patients don't have an immune system that are responding, so they're missing the entire immunopathology aspect. They just are having, what does TB do? As far as direct damage effect, so maybe lung infection or disseminated infection, and that's straight up what damage is TB doing? MAC and pneumocystis. Now what happens when we initiate HAART treatment on patients, we bump up their immune system. And now all these immune cells are in the midst of massive infections of TB and MAC and pneumocystis and all these other guys, right? And so they're sitting there going, whoa, where did all this come from? Now it's pretty much, just think of it as this person had underlying sepsis. And we now introduced a strong immune response in someone that has sepsis. Guess what happens? It worsens. It gets worse. We have strong immunopathology plus the pathology from the infection itself. And so that actually is what happens after HAART initiation in these patients. And so the factors that determine how bad IRIS is pretty much are how suppressed were the patient's immune system before we gave them that HAART. And then how quickly did that immune system ramp up after we initiated this? And how low did we bring our viral load, right? So those sorts of things kind of interplay on how quickly our immune cell respond or grow back and then respond overload to where there's strong immunopathology, and this could progress this person's all sorts of damage from whatever the infection they had underlying. And so what actually happens as far as the cellular level is a rapid increase of CD45 Rho positive T cells. These are pretty much your memory T cells. So your memory T cells are these 45 Rho positive T cells. Those are your memory cells, and so we see a rapid increase in there. And then later on, we see a second slow increase in T cells, and these are because we're making new naive T cells in our thymus, and those are your CD45 Ra positive and CD62 L positive naive T cells. Now you're thinking, oh gosh, more CD markers. I already learned a ton in immuno. It's not really important to know the CD markers here. I mean, it's maybe nice to be able to recognize in case they ask you that detail, but usually they're more asking questions. Do you know that the initial is memory T cells that bump up, and then the second slow increase is just simply naive T cells responding? And this rise can lead to inflammatory response because this person might have an underlying opportunistic infection floating around, and now our immune cells are working, and that can lead to immunopathology, as we have discussed. And so that pretty much ends the module on HIV and AIDS. Thanks for watching, and don't forget to subscribe and like, and subscribe to the channel as well. And we'll be discussing our CNS module coming up hopefully this weekend or next week. That should be quite informative and fun for you guys. So I wish you the very best and happy studying. And if you have any questions, post on my Facebook page or even here on the YouTube video as well.